During the 19th century, an Austrian monk named Gregor Mendel conducted experiments in his garden with pea plants, leading to the discovery of the basic mechanisms of heredity. Thus, the science of genetics was born, and humanity took its first small steps toward deciphering the genetic code. First, some basics. An allele is one pair of genes that appear on a particular location on a particular chromosome and controls the same characteristics such as eye color or skin tone. So basically, it's one part of something called a genotype. A genotype is a combination of alleles situated on a corresponding chromosome that determines a specific trait. So say you've got the genotype for freckles, which is a dominant gene, meaning it's more likely to have freckles than no freckles. Your genotype will look like this, or like this. See that little f? That's a recessive allele, meaning that if a dominant allele is present, then the recessive allele will be overpowered and will not be shown. Now let's go back to our genotypes. You see how there are two different genotypes I just showed you for a dominant gene? This one is called a homozygous genotype, meaning it has two of the same allele, which is whether it's dominant or recessive based on the gene being expressed. The next one is called a heterozygous genotype. That means that one allele is dominant and one is recessive. Homo, same, hetero, different. So we covered what a trait looks like when it's being written down. Now let's talk about what it actually looks like. A phenotype is a set of observable characteristics of an individual resulting from the interaction of a genotype with the environment. So if we go back to our genotypes, say you have a homozygous dominant genotype for freckles. This means that the phenotype of the gene would be freckles. So if you flip the script and say that you now have a homozygous recessive genotype for your freckles, the phenotype would be no freckles. Still with me? Let's move on. We talked about how a gene is expressed and how it looks. Now let's talk about how we determine how each gene is passed down from parent to child. During the early 1900s, a man named Reginald C. Punnett was doing Mendelian experimentation on plants and animals at Cambridge University. This led to the development of the Punnett square, which depicts the number and variety of genetic combinations. The Punnett square quickly became the easiest way to plot the passing of genes through generations. First off, a Punnett square looks like this. Let's go back to our genotypes. So say you want to determine the possible combinations of genotypes if you were to have a child with someone with no freckles. No problem. So say for the sake of our example, you have a heterozygous genotype for freckles, meaning your genotype looks like this, and your lovely spouse has a homozygous recessive genotype for freckles, meaning their genotype would look like this. Let's go back to the Punnett square. Using the Punnett square is simple. You put your genotype along the top and your spouse's genotype along the left side. All we have to do now is mix the genotypes together like this. This shows the possible outcome of when you have 4 children. Looking at the new genotypes, we can see that half of the genotypes are big G, little g, and the other half are little g, meaning that there is a possibility that half of the children will have freckles and the other half will not. Moving on. Mixing the different genotypes may seem easy, but it's not always that simple. Sometimes 2 alleles of a gene pair in a heterozygote are both fully expressed. This is called codominance. Say we're talking about the stripes and dots on a turtle's shell, and say that if a turtle with only dots on its shell were crossed with a turtle with only stripes on its shell, there's a possibility that the offspring could have both stripes and dots on its shell. How would this happen? Let's find out. Going back to the Punnett square again, this time let's put the genotype big S for stripes on top and big D for dots on the left. When we cross these together, we see that all the possible genotypes are heterozygous and they look like this. The genotypes are heterozygous, but both the allele for stripes and dots are both equally expressed, meaning the shells of the turtles with the genotypes SD would both be striped and dotted. Make sense? Let's talk about what would happen if one allele does not completely dominate another. When this happens, it's called incomplete dominance. For instance, if we have a flower that is homozygous blue and we cross it with a flower that is homozygous red, the possible outcomes would be a new phenotype. Let's go to the Punnett square. This time, we're going to put the genotype R for red flower on top and B for blue flower on the left. When we cross these, we see that the genotypes are heterozygous and look like this. In this example, instead of resulting phenotype being a combination of the 2 alleles being both equally expressed, we get a completely different phenotype. So instead of a flower that is both red and blue, we get a purple flower, which is a mix of the alleles where neither is more dominant than the other, resulting in a new phenotype. In this video, we went over alleles, genotypes, dominant genes, recessive genes, homozygous genotypes, heterozygous genotypes, phenotypes, Punnett squares, codominance, and incomplete dominance. Thanks for watching my attempt to teach a 9th grade class basic genetics. I will never be a teacher.